Good morning or good afternoon, everybody. I do not have any conflict or interest, but I always like to start my talks showing the impact of this epidemic, this HIV epidemic, which is still a silent pandemic that we have in the world. And we already had like more than 35 million people died. One of them was Frank Moore, was the painter of this beautiful painting here. And we still have the HIV infections happening every year. More than 1 million infections every year and a lot of deaths. So it is interesting that we've been focused a lot in the use of immunotherapy for a lot of fields, for instance, for cancers or for subtypes of immune diseases. And among the 10 of the most advertised drugs in 2019, 3 of them were monoclonal antibodies. One of them against migraine, the other 2 against autoimmune diseases. And we have other antibodies that have been used now for pathogens, some of them for like bacteria, like for C. diff, and even for virus for the RSV. So it's not a surprise that scientists are trying to use the same products against HIV. For the past more than 10 years, we've been finding and developing HIV-specific neutralizing antibodies or broadly neutralizing antibodies. I want to call them bNAbs. And here I can show them all the bNAbs that have been used already in clinical development and evaluated in humans, all of them in red. They can be divided according to where they bind in the spike of the virus. So we have the CD4 binding site antibodies, like those here, that bind in this area. We have the V3 glycan site antibodies, the V1, V2 glycan loop or site antibodies, and the AMPER part, which is this intersection between GP41 and the cell membrane or the external part of the virus. So we have several products already being evaluated in humans. And we also have products that were genetically engineered based on those antibodies. We have a tri-specific that has like 3 FABs, each one of them binding to a different area of this spike. And you also have bifunctional antibodies, one of them that binds to the one area here, the AMPER area, plus another FAB part of the antibody that binds to the CD4 receptor itself in the cell. So we have a plethora of types of antibodies that can be used to be evaluated in humans. When we talk about antibodies, we always think about 2 words, breadth and potency. How many strains of HIV they can cover, and what's the concentration that we're going to have to use to neutralize those viruses. So here in this graph, we have a distribution of those antibodies, several of them coming from that previous slide. So we were expecting the antibodies to cover a lot of strains, like here, 100% of the strains, in a very small concentration, or is a very low concentration or high potency. So the ideal antibody would be here in this area, would be like the gold standard for us. Of course, we do not have this molecule yet. And we can represent those antibodies also in another type of graphic using a panel that we have here at the VRC that uses 208 strains of HIV that we can test against each one of those antibodies and see how many of them get neutralized. So the more potent we have here, the more to the bottom. And you can see here on the top how many strains are, or the percentage of strains that are not neutralized by those antibodies. So you can see the sum of antibodies, like for instance, VRC07 or N6, they are very broad. They do not neutralize a small amount of strains, but the potency, although it's very good, is not as good as, for instance, Cap256, which is a very potent antibody, but does not neutralize a lot of strains. So what we're doing now is trying to find ways to improve those antibodies, because one antibody at this point is not going to be enough for prevention or treatment. We tested VRC01 and 3BN-C117 in several participants with HIV, and we noticed that once we stop giving the antibody, after some time the virus rebounds. And here we see that one of the reasons that they rebound is because of resistance. It is logical because the HIV is highly mutated and is always forming new strains trying to fight against antiretrovirals, and this is a very important antiretroviral. So among the strategies we have to improve the bNAb therapy, the first one would be like to combine 2 or 3 antibodies. Here we have some heat maps where they show how those antibodies fare against that 208 viral panel that I showed in the beginning. Here we have the VRC07, 523LS. It covers a lot of strains. The darker here, the redder here, the more potent it is, and in blue, it doesn't cover very well. So we have PGT-21, PGDM-1400. Also, they cover a lot of strains, but when we combine them, then it's even better. The problem is that we do not have a lot of strains that are sensitive to all the 3 antibodies. We have a lot that are sensitive to 2 antibodies, but even of them, we're going to have some strains that are going to be against only 1 antibody, even like in monotherapy. We should have like a combination that covers as much as we can to prevent resistance. And the other problem that we have is that this does not distribute very equally among the clades. So you can see that this combination of VRC07, PGT-21, PGDM-1400, it covers very well clade C and clade B, but does not cover well clade AE, which is circulating in Thailand. So we're going to need like to find new antibodies that can combine with the other 2 antibodies to form a better cocktail that you can give to people that can cover all these strains. We're still like working those antibodies. Every month we see like people developing those new compounds. What we can do is to improve the breadth and potency, use the knowledge you already have in our hands. Here at the VRC, we are using structure-based rational design to change some residues in the molecule to improve the breadth and potency, or use a surface matrix screening to also do the same thing. So that, for instance, a good example that we have is TENI8. When we have TENI8 in our hands, was a very good antibody, a very broad antibody. It does not cover 2% of the strain, so it's very broad, but it was not very potent. But through those modifications, we were able to increase 20-fold the potency of this antibody. So you can see that like by the knowledge that we have now, we can improve those antibodies, and we're doing that now with the VRC in combinations with other institutions. Another thing that we can do is to improve the serum half-life of those antibodies by adding some mutations in the Fc part of those compounds. Some of those mutations have already been added. We have, the VRC has added, and other groups have added the LS mutations. They increase the ability or the avidity to the FcRn receptor, which is the receptor that helps you to recycle those antibodies through the endothelium. So by improving or by increasing the avidity, we increase the potential of this circulation of the antibody in the serum. We already gave like combinations, not combinations, or antibodies that had the LS mutation and did not have the LS mutation. In our case, we have the VRC01. You can see here in this graph that the VRC01 itself in black, here you can see that in 10 weeks is already very low. But when we give the VRC01 LS, we can see that even like smaller concentrations, it can keep in more than 20 weeks in a very good concentration in the plasma. If you give like 40 milligrams per kilo IV, then you can have a very high concentration here, like 100 micrograms per ml for more than 25 weeks. So it is very exciting that we have those molecules, and we have studied that even like how those molecules fare in the mucosal of participants that receive those molecules. So HVTN 116 compared VRC01 and VRC01 LS in the distribution of vaginal and rectal mucosa, and we observed that VRC01 LS could be detected in those mucosal samples 12 months after administration, which is a good thing for prevention. Another thing that we can do with those molecules is improve the effector function. So at this point, we're talking only about virus neutralization, but we know that antibodies have other functions. For instance, it can have ADCC or antibody-mediated cell cytotoxicity. When the cells start expressing those antigens on the surface, the antibody can bind there and can help to bring macrophages and NK cells to kill those cells, either by ADCC or by phagocytosis, or even by complement. So by changing also the Fc part of those antibodies, we can increase the avidity to Fc receptors or decrease the avidity to Fc receptors. We published a paper last year where we changed the BRCA07 factor 3LS. We added some mutations called DEL mutations and other mutations called LALA mutations. The DEL mutations increase ADCC and LALA mutations decrease ADCC. And we saw how that can change the profile of those antibodies in non-human primates. Of course, in some cases, we're going to want to have the ADCC improved. In some cases, we don't. So we can play with those molecules to fit how we want this molecule to work. We can also genetically engineer those molecules, as I told you in the beginning. Sanofi, in combination of the VRC, formed a compound called SAR441236, and it has been tested now in a CTG network in a trial 5377, where we evaluate the PK and safety of this molecule. This molecule has 3 FAB parts, one FAB for VRC01, the other one for PGDM 1400, and the other one for 10E8V4. So it's one molecule with 3 FABs. If given individually, we see that the virus rebounds very easily, but when we give this combination, we do not see the rebound. So showing that this molecule is a very good tool to be used in humans. We know that it's very safe at this point, but now we still need like some results to show how the PK and how efficacious it is when HIV infected or even in prevention. Another thing that we can do is to do like bifunctional antibodies, like this molecule that was developed by David Ho's lab, in which in one part of the antibody, one FAB, it is against the envelope of the virus, and the other one attaches to one of the receptors that is used for viral entry in lymphocytes. So in this case, we have a molecule that has the 10E8 V4 in one arm, and the other arm is the imAb, which binds to the CD4 receptor itself in the cell. And with this, it kind of brings the antibody closer to the membrane of the cell, increases the chances for the other part of the antibody to find the virus when it's budding from those cells, leading to ADCC or neutralization. And we already have tested this antibody in humans in the trial AAAS1239 with very good results and safety. One very exciting technology that we still have to improve is the vector technology for delivery of those antibodies. A good example is the Miami monkey that was published some time ago by Rhonda Roger's group in Florida, in Miami. So that group gave 3 AAV vectors, like AAV is a virus, a parvovirus, that can carry the DNA to express those antibodies. They gave those AAVs to one monkey. In fact, it was like 3 AAVs. One expressed 3BNC117, the other one 1074, and the other one, if I'm not wrong, 10E8. The 10E8 vector did not express, but the other 2 vectors expressed very well. And the monkey received those viruses, expressed the antibody, as you can see here, and the virus was suppressed. And if it's still suppressed after several months, and if I'm wrong, even like after years. So there's a very good potential to be used in humans. And the VRC is using this vector AAV8 VRC07. We're testing the safety and the PK of this product in humans in our clinical center here at the VRC. And we're going to be publishing those results soon. And as we saw with the COVID epidemic, mRNAs can be also used to deliver the message for the production of proteins. We have companies that are collaborating with the VRC for the development of mRNAs that can bring the molecule to express antibodies, to express the proteins instead of like vaccines. This is still in the beginning. It's going to be complicated because in this case, we're going to need like 2 mRNAs or a very long mRNA to express both arms of the antibody and to make sure that they kind of pair perfectly. But it's a very nice technology to be developed in the future. And what are the target populations for the HIV bNAb use? The first thing that comes to mind is prevention. In fact, we had a trial run by HVTN, HPTN networks, the AMP studies, where we evaluated the efficacy of BRCA1 in preventing HIV infection in African women and MSM transgender men in North and South America. More than 4,000 participants went to this trial. Some received placebo, some received 10 milligrams per kilo for BRCA1, and the other one 30 milligrams per kilo, given every 2 months. In the end, the VRC produced more than 80 kilos of antibody for this trial. You can see that is a very large amount to give into this efficacy trial. The results were quite good, in fact. I mean, we noticed that the VRC was effective at preventing the acquisition of sensitive HIV strains. The VRC1, however, did not prevent acquisition of resistant HIV strains when they have the IC80 of more than 1 microgram per ml. We shot that in another number, thinking that a lower concentration would be able to prevent, and it didn't work. So we're going to have to use more antibodies or a higher concentration of antibodies for prevention in the future. But it gave us some information for the potential to use the antibody given SC or subcutaneously instead of IV, and also brought an incentive for the commercial development of this product. I can see this as a wonderful tool for the prevention of mother-to-child transmission. So if you have a mother that is HIV positive, and as soon as the baby is born, you can give this antibody or a combination of those antibodies and protect during breastfeeding. That's a wonderful tool that you should explore more. And it's being explored, in fact, by the Impact Network. We have several trials when they test the safety and efficacy, not efficacy, but the safety and PK of those antibodies in infants. Another possibility is to use those antibodies for the treatment and for the HIV eradication. So we have already trials testing bNAb combos or in combination with long-acting antiretrovirus. For instance, the CTG 5357 is evaluating the efficacy of a combination of cabotegravir and VRC07-523LS given every 2 months, monthly for cabotegravir, 2 months for VRC07-523LS, or the combination of several antibodies. The BIDMC group in Beth Israel, they are giving VRC07-523LS, PDT 2021, PGDM 1400 to see both HIV patients that are viremic or suppressed. And as I told before, the A5377 that is testing the tri-specific SAR441236 in HIV infected patients, also viremic and suppressed. We are testing now also those products in pediatric strategies for the Impact Network, like in P2008, P115 with VRC01, and even the combination of 2 antibodies with the Tatello trial by the Harvard group with VRC01-LS and 1074 in children. We also are testing, or several groups are testing, bNAbs in functional cure and HIV eradication, especially for the kick and kill strategy, where we're going to wake up reservoirs using the kick product, and then the antibody is going to be the kill for this kick. So we're going to give a latency reverse agent in the presence of the antibody to improve the immune system. And with this, we're going to have the increase of ADCC and killing of those reservoirs. Several trials are now in progress testing this combination with HDAC inhibitors, with some of those antibodies, or with other immunotherapies, so for instance, like interferons or IL-2 or TLR9 and TLR7 agonists. So we're going to have results from those trials and see if this combination is going to really be efficacious against the reservoir, decreasing the number of the reservoir in those patients. Another possibility is to have an vaccinal effect of those bNAbs once they form immune complexes with the virus. So we had a study by Mal Martin in which monkeys were infected with a SHIV, and then 3 days later received a combination of 2 antibodies, C117 and 1074, and he and his group observed that several of those monkeys, after the antibody was down, the virus didn't come back. In fact, the virus only came back when they depleted the CD8s, meaning that it's possible that giving antibodies in the beginning of the infection can help with this vaccinal effect. Michel Nuzenzweig also and his group also saw something similar in humans, so it is a very exciting possibility that can be explored in the future. So in summary, we have more than 17 bNAbs or related products in development for various applications like prevention, cure, kick and kill, and vaccinal effects. We already have results from the AMD study showing that it can be useful for prevention and also dictating how those molecules are going to be important for the future. And if you think about that, it is a new tool that you should be using. It is a very safe tool that should be using to fight the epidemic, and novel products, not only in terms of one molecule, but forming trispecifics and bispecifics, and also vectors for the delivery of those products are in development. And we're going to have a very exciting results in the next 2 or 3 years that are going to be very important to be discussed in these panels as well. I would like to acknowledge all the VRC group and all our collaborators that have been testing and evaluating our products in extramural trials. Thank you.